Your normal B12 level is melting your brain. How could that be? How could a normal lab result for vitamin B12 lead to brain melting outcomes? That's what we're going to talk about today in looking at a study that was just published in January in the Annals of Neurology. And what you need to understand is that what I've taught for years and what functional medicine teaches is that normal lab ranges are not healthy lab ranges. There's a difference between normal and healthy. In this paper, I hope drills it home for you to the point where you see that functionally low vitamin B12, this study shows functionally low levels. So levels within the normal range, but that are not optimal, promote mild cognitive impairment, which is the precursor to dementia, and therefore also promote dementia. Before we dive in, let's look at this picture here because I put these pictures up here for a reason, not just to stump the viewer. So if we look at the picture here, this is a picture of the inside of your brain. And what we can see here, this big yellow cell here is called an astrocyte. And astrocytes are cells inside your brain that you can see with its feet here, it's holding on to two separate nerves. Okay. And nerves are made up of the axon, which is the long, skinny part that runs along it. And then on one end is a body with the cell nucleus that conducts a signal or receives signal this way. And then at the other end is the end that's going to conduct the signal to the next body of the neuron here. So you have the synapse where it releases, and the next body of the neuron takes it and sends it that way. And you'll see that you have myelinated nerves, which is fancy for nerves covered in myelin. It's this blue circular kind of roll around the axon. Well, then the myelin is what allows your nerves to conduct signal quickly. Okay, we have myelinated nerves, which are ones covered in the myelin. And then not shown here is that you can also have nerves that are unmyelinated. So they'd just be a nerve without the blue rolls on it. Okay, let's draw that so that it stays up there. So here's an unmyelinated nerve. Okay. And so the difference between these two is the myelin. As you can see, one has the blue roll, one doesn't. But the difference goes beyond the myelin. What the myelin allows you to do is conduct the signal faster. Myelinated nerves conduct signals faster. Unmyelinated nerves are slower. Why? Well, because say you have a signal here and it's going to conduct down this nerve. Well, it's got to travel the entire nerve. Bing, bing, to the end, right? Myelinated nerves are allow the signal to conduct faster. It starts here, and then here, let's use this one up here, right? So if the signal started here, it goes boom, boom, boom. It gets the skip areas, right? So the myelinated nerves, the signal travels a lot faster than the unmyelinated nerves. And that's important because we want to send certain signals quickly and certain signals not as fast. Unmyelinated nerves are also called C fibers. Okay. And C fibers are the nerves that conduct pain. So when you are in pain, these fibers are firing constantly and the signal travels slowly. Okay. But the myelinated nerves are called A fibers. And A fibers conduct things quickly, as we just said. The signal will go bing, bing, bing. So you've all experienced the difference between these two nerves. How so? Well, if you've ever skinned your knee when you were a child, skinning your knee fired the C fibers. Oh, mommy, my knee hurts, right? And what's mommy do? She comes over and she rubs your knee and she says, "It's okay, baby. You'll feel better." And by rubbing your knee, she's firing the A delta fibers, which are fibers that outcompete the slow pain signal to the brain. So the slow pain signal is going from your knee up to your brain, Whereas the rubbing of your knee, or if you move your knee, any movement or friction or rubbing vibration, bing, 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 traveling quickly to the brain. So A fibers outcompete C fibers to the brain. So mommy coming and rubbing your knee, whether she knew she was doing it or not, she was that rubbing sensation or the movement or vibration, the activation of those nerve endings were outcompeting the pain to the brain, and now your knee doesn't hurt as much. Okay, so myelin is very important for the myelinated nerves. When you lose myelin from a disease standpoint, that could cause neuropathy, that could cause multiple sclerosis, that could cause ALS, that could cause various disease states when we lose myelin. And vitamin B12 is crucial for myelin production. If you don't have enough B12, you're not going to make sufficient myelin, and now you start to have neurological symptoms because of that. And what symptoms do you have? Well, that depends on what the nerves that are losing the myelin control. Okay, so which area of the brain is losing its myelin depends on what kind of symptoms you have. So with that context, let's dive into the study here and see what we can learn. So the study we're going to cover today, as I said, was published in January of 2025. So hot off the presses here, published in the journal Annals of Neurology. And the title is Vitamin B12 Levels Association with Functional and Structural Biomarkers of Central Nervous System Injury in Older Adults. We're going to go over to the actual PDF version of the paper so we can dive into the details. All right, so here we are on the actual paper. And again, this is very important for anyone who wants to have a healthy brain over the long term. So if we dive into the guts of the paper, all right, what they did is, well, first they say B12 is crucial in production of fatty acid, amino acid, and nucleotide synthesis. B12 is crucial in fatty acid and amino acid metabolism and production of nucleotides. The exact threshold for adequate levels of B12 remains undefined in terms of functional impairment and evidence of injury. The objective of this study was to assess whether B12 levels in the current normal range in a cohort of healthy older adults may be associated with measurable evidence of neurological injury or dysfunction. So to test that, the authors took 231 healthy elderly volunteers, average age 71, with a median B12 blood level of 415, we'll call it. Okay, so we have in these 70-plus-year-old adults, we've got their, let me make some space here so we can scroll down. Okay. We've got their B12 levels here. If we say, put your quote-unquote normal range here from your Labcorp result for B12, you're going to see from around 200 on the low end cutoff to about 1100 on the high end cutoff. Okay. So in this study, the median range for the elderly people, we'll call it, is 415. It was 414.8, but we'll call it 415 for easy communication. Okay, so we performed multiple ways to assess the nervous system. They looked at visual evoked potential testing, so some vision stuff. They looked at processing speed cognitively. They looked at MRIs of the brain to look for white matter lesions. And they also looked at blood markers that would show us that there was nerve injury, astrocyte involvement, or amyloid pathology related to dementia and Alzheimer's. So if we dive in here, we're going to get really neat information. So let's go a little bit further down. Cobalamin is the other name for vitamin B12. So we could say B12 or we could say cobalamin. You're saying the same thing. Vitamin B12 is an essential vitamin normally acquired through absorption in the enteric system in mammals. So basically, we get B12 from eating food. Deficiency in humans is caused by a lack of intake, which includes a vegan diet, because a vegan diet is not eating animal products, and animal products are the best source of vitamin B12. You could also have a deficiency due to disease, say stomach issues, because the primary absorption of B12 is going to be performed by intrinsic factor binding to it and absorbing it from the stomach. So disease or procedures in the digestive tract impairing absorption. So if you've had a stomach procedure where you had your stomach stapled or a stomach sleeve, and you're decreasing the surface area of the stomach, you're decreasing your production of intrinsic factor and absorption of B12, and that could be leading to this. Or if you've got something like celiac disease or a small intestinal pathology that is damaging the villi and you're not absorbing things that way optimally, that can also contribute. So after depletion of B12 stores, signs of deficiency such as hematological or neurological symptoms may develop, including megaloblastic anemia and subacute combined degeneration of the spinal cord. So in other words, if you have low B12, okay, low B12 equals change in your cells. So specifically, your red blood cells get too big, and that would be evidenced by high MCV, MCH, MCHC on your CBC. Okay, so if you're getting high or red blood cells that are too large, indicated by these markers being high, that can be megaloblastic anemia or B12 anemia. And so, or pernicious anemia. So if you lack B12, you see those cellular changes. You can also see spinal changes and nervous system neurological symptoms, such as ataxia, which is a lack of coordination. Okay, so maybe now you stumble a lot, or you bump into walls more than you should. Your spatial awareness is off. Okay, you might also have paresthesias, which is basically numbness and tingling. Okay, so numbness and tingling in the face, the hands, the feet, and or weakness. All right, so neurological symptoms like that are classical dorsal column symptoms, where if you're not having enough B12, you don't have enough myelin produced in the spinal cord. Those dorsal columns of the spinal cord can be impacted, and you develop these neurological symptoms potentially. So again, B12 is important for maintenance of intact myelination. If we don't have optimal B12, we don't have optimal myelin, and we may have these neurological symptoms. We may also have the cellular symptoms tied to the red blood cell size. In addition to affecting general cognition and memory, vitamin B12 deficiency may lead to dementia and psychosis, suggesting a broader dependence of the brain on B12. Again, why might we have dementia and psychosis? Well, because if we don't have optimal B12, we're not optimally producing myelin, and the conduction of those nerves isn't happening optimally. So now that could be psychosis, that could be dementia, that could be movement disorder. The VitaCog study established that B12 supplementation, that B vitamin supplementation, including B12 in older adults with mild cognitive impairment, leads to both functional and structural benefits. So is it useful to take B vitamins? You know, your medical doctor, when they test your blood, if it's lab high, okay, if you're greater than this 1100 here in the blood, a lot of times they tell you stop taking your vitamin. Well, it might be high because you took your vitamin. B vitamins are what are called water soluble. Okay, so it's a lot harder to be toxic in B vitamins because they're water soluble. And the research says that typically they reach their peak 1 hour after you take them in the blood, and then you're spilling them the rest of the day. Okay, in other words, if you've ever taken a multivitamin with a lot of Bs in it, or a B complex with a lot of Bs in it, and your P became neon yellow or neon green, that neon color is the B vitamins that you are spilling. So, you know, before you stop taking your B vitamin supplement because your doctor, you know, doesn't know nutrition maybe as well as they should, or because they're just against nutrition and they say you don't need that. Well, there's multiple studies showing that vitamins preserve brain health and slow cognitive decline in older adults. Many studies from many different research groups in many journals, and we've covered multiple of them on this channel. B12 supplementation slows the progress of brain atrophy and mild cognitive impairment and of clinical decline in multiple areas of cognitive testing. Boom, right there. Moreover, B12 deficiency is associated with a higher burden of white matter hyperintensities in the brain. In other words, you have more white matter lesions on MRI. Well, those things are not good. Those can be associated with migraine. Those can be associated with multiple sclerosis. Those can be associated with multiple different brain pathologies. We don't want to have too many white matter hyperintensities. And here's another one. B12 deficiency is something that can cause it. Those white matter hyperintensities could indicate microstructural changes reflecting overall brain health. One of those is chronic ischemia or not enough blood supply. Look at this red box. This is key here. In the United States, the cutoff value for B12 deficiency is currently defined as below 148. Okay, so that would be down here even. Okay, so B12 deficiency is even lower than what Labcorp says. Okay, this value was simply calculated as 3 standard deviations below the US population average, independent of clinical observations. Okay, so how are these normal ranges set? Not by clinical observation, not the B12 one, right? They just did 3 standard deviations from the average. Well, if we use this study, the average was 415. So 3 standard deviations would bring them down here. And so the threshold for health is very low. And that's why we say health in quotation marks, because you are not healthy here. You're not healthy here, as we'll see. Okay, so going by the quote unquote normal range is laid out by Labcorp or by doctors that don't understand health is dangerous. The American Society for Nutrition criticized this approach in 2010, arguing that more than 5% of patients who have a syndrome consistent with B12 deficiency and who respond to B12 supplementation have blood levels above that threshold. Okay, so this threshold here, the American Society for Nutrition said that's terrible. Okay, because more than 5% of patients have levels higher than that are showing symptoms of B12 deficiency. And when we give them B12 supplementation with levels up here, they do better. So there's no way that this can be a good level to use for our cutoff for health, right? All right. Other studies demonstrated that B vitamin supplementation was beneficial in people with clinical features of B12 deficiency, regardless of the measured levels in the blood. Okay, so there's some studies showing that regardless of what your level is, if you have symptoms of B12 deficiency, B12 helps. Okay, so what if someone had an 1100 or what if someone had a 1200? They were actually lab high, but they had B12 deficiency symptoms. There's studies that say give them more B12, all right? Because there's studies that show people benefit from it, and we'll show you later why that might be the case. Selecting a cutoff value based on clinical observations would better reduce disparities in B12 deficiency diagnosis and management. Cases of biochemical B12 deficiency, where suboptimal B12 levels have been reported without overt clinical manifestations, have been called subclinical cobalamin deficiency, or you and I could say functionally low B12. All right, so they're saying we see people with B12 deficiency symptoms with functionally low B12 levels. So again, we need to do better with what our normal ranges are. Subclinical B12 deficiency is most prevalent in the elderly and is associated with greater white matter hyperintensity burden. All right, so let's move on here. Defining thresholds for optimal B12 levels based on clinical findings is crucial. So again, we want to go by clinical findings, not by an arbitrary range on a piece of paper. Recording visual evoked potentials is a validated and non-invasive tool to assess myelin function in the visual pathway. So that's a test they use in this research study. And they tell us a little bit about B12 physiology here. Once B12 is absorbed, the transport proteins haptocarin and transcobalamin bind it with great affinity and act as transporters in the blood. Since transcobalamin, or TC, it has a specific receptor for cellular uptake, hollow TC is usually considered to be the biologically available form or active form of B12. On the other hand, hollow HC is the not immediately available form or the inactive form of B12. We hypothesize that lower total B12 levels within the specified normal range may still be associated with subtle functional and structural neurological deficits if the active fraction of B12 is too low to sustain adequate cellular levels. To that end, we evaluated the association of B12 concentrations with markers of myelin integrity, the visual evoked potentials, cognitive performance, blood biomarkers, and MRI. Another interesting note here to me was blood levels of the hollow TC, which is the active B12, methylmalonic acid, which is a great marker for B12, a better marker than serum B12, and homocysteine were all within the normal range for these people. So not only the B12, but these other three markers for B12 were all in normal range for these people, but they all had neurological deficits. Okay, so that's very important because again, if everything looks normal and people are saying, I have neurological symptoms, a doctor might say, oh, it's all in your head. Here's a psychiatric medication. And that's dangerous for two reasons. A, you're giving them a drug they don't need, so that's 100% risk. And B, you're not addressing their issue, which is a deficiency in a nutrient that's needed for global physiology. And so they move forward and continue to get worse. So interesting to know is that this study was done at the University of California, San Francisco Memory and Aging Center. And B, all of the 231 participants did not have history of neurological or systemic conditions. So that's important because we want to be able to say, is there a neurological issue due to B12, lack of B12, or some other diagnosis they have? They don't have some other diagnosis. So you can more confidently say it's the B12 tie-in. Higher scores on the visual evoked or on the cognitive performance testing indicate slower processing speed, so worse performance. So the results, 231 healthy elder adults had their total B12 levels measured, which had a median of 415 picomoles per liter, and a range of 282 to 557 in this group. Patients with lower levels of B12 showed a significant inverse association with the visual evoked potential latency, basically meaning they had slower conductivity in the brain. Okay, so lower B12, slower conduction. Lower B12, less myelin, slower conduction. Right? Lower B12, skin your knee, mommy rubbing it doesn't help you as much. Right? Slower conduction. We don't want slower conduction. We want fast conduction. The lack of available B12 induces delay in conduction speed through the brain. So again, we must have the B12 to have the optimal conduction. Overall, these results indicate that in a healthy population with B12 blood levels within the currently defined normal range, there's an inverse relationship between the biologically available B12 and the latency of visual evoked potentials. Okay, so the lower your B12, the slower the cognitive function, the slower the conduction. Previous studies show B12 deficiency can cause cognitive impairment. How about in this study? What did they find? They found that higher B12 related to faster performance, lower B12, slower processing speed. On MRI, B12 deficiency is associated with white matter hyperintensity, suggesting possible microstructural tissue disruption or increased vascular risk. Okay, lower B12, more white matter lesions, greater vascular risk. What's a vascular risk in the brain? Stroke. All right, let's get to the meat of it here. More meat? Yeah. The participants with the highest white matter lesions had hollow TC in the lowest range. So the highest white matter, lowest B12. These analyses suggest that even within the currently defined normal range, lower levels of available B12 associate with larger volumes of white matter lesions. So again, if that doctor sees your B12 at a 400 and says, hey, your B12 is normal, that doctor is promoting development of white matter lesions in your brain melting. And that's not necessarily that doctor's fault right now because all the normal ranges are wrong, right? Based on this study. Personally, practicing functional medicine for years, I've said anyone below 600 is functionally low or not good. So we've been helping people a lot sooner, right? So you want to be above 600. This study's showing 400 and less is not good. Our results suggest that in a cohort of healthy elderly individuals, lower B12 associates with a delay in visual evoked potentials or brain conduction and that it is specific to the biologically available fraction of B12 bound to transcobalamin. Low levels of B12 associate with slower spatial processing speed in an age-dependent manner. On MRI, participants with lower B12 have higher white matter intensities. Remarkably, we find that high levels of the biologically unavailable fraction of B12 associate with an increase in the levels of tau protein in the serum. That's a biomarker for neurodegeneration. We've talked about this in previous videos. The tau protein is one of the blood tests you can run to look for dementia risk. And remember that blood test was better than dementia specialists at properly picking out the people who are on their way to dementia. So if you have high levels of biologically unavailable B12, you have more tau, and that's a biomarker for neurodegenerative risk or dementia risk. These findings suggest that current parameters for defining adequate B12 levels may be inappropriate when considering neurophysiological, neuropsychological, serological, and neuroradiological outcomes. In other words, we need to change our ranges because we're failing patients. B12 deficiency results in prominent white matter injury of the posterior columns in the central nervous system in humans. Again, the posterior columns control your balance, your coordination. The necessity for sufficient blood levels of B12 to maintain nervous system health, particularly myelinated tracts, is huge, right? Low B12 and high homocysteine can have profound impacts on the brain. Our study has the additive value of showing a precursor stage at which microstructural changes are present and already impairing neurological function. So as a functional medicine clinician, my goal is to find things as early as possible, right? Well, that's where the real value of this study comes in, because they're saying right here, we're showing a precursor stage. Our results have the value of showing a precursor stage, or the levels of B12 that are not good so early that it's impairing neurological function before medicine will diagnose it. So they're saying here, what's that precursor stage? Well, below 415 is not good. Where, medicine's saying you got to be below 200 or some areas below 148 before they call you deficient. So we can find it sooner if we adjust our range and make it tighter based on health and based on science here. In addition, older participants in our cohort exhibit a functional correlation with involvement of low B12 and myelin integrity. So again, lacking B12, lacking myelin. We think that the observed detrimental effect of low B12 on the central nervous system is related to myelin integrity. Metabolic syndrome and in particular insulin resistance have been linked to biomarkers of low B12. Okay, so how do we dial this home to everyone out there? You might say, well, I don't have mild cognitive impairment. I'm mentally clear. I don't have symptoms of stumbling into walls and things like that. Well, not yet, right? So I don't have to worry. I don't have that stuff. Yeah, but most of the population in the United States is overweight. Most of the population has issues with blood sugar handling and insulin sensitivity. And this is saying right here, insulin resistance is linked to biomarkers of low B12. So those two are associated. And look at this. Right above, we skipped over it, but it says right here, no participant in the study suffered from severe obesity. So you don't have to be obese necessarily for it to be impacting or related to your B12 level. So we want to have good B12. We also want to have good insulin sensitivity, good overall health. The plaques seen in Alzheimer's disease contain tau proteins, and B12 plays a role in preventing these proteins from aggregating. Therefore, the retention of B12 in the inactive form in the brain favors tau-mediated neuronal injury. So we need the active form to help fight the dementia. Now, we said earlier, we would get to a point where different B12 levels are different for different people. Well, why might that matter? Well, there is genetic variability to B12, right? Why? Well, because the genes control the proteins that bind B12. So you can see there's transcobalamin 1 and transcobalamin 2 genes, and these affect the levels of B12 in the blood because they affect how well you're binding the B12. Okay, so I might take one dose, you might take another, and we will both have different B12 levels because our genes may be different. So there is some genetic or epigenetic play there. Genetic and epigenetic. Our findings are of critical importance for rethinking the biologically sufficient B12 levels. The population-based studies that defined healthy micronutrient levels may have missed the subclinical manifestations of low or high B12 at the extremes of the population distribution that can affect people without causing symptoms. Our findings support the idea that subtle neurological deficits manifest at higher levels than the current threshold defined for deficiency. And most importantly, it could provide an explanation for the often reported discrepancy between hematological and neurological symptoms. In other words, our findings support the idea that subtle deficits or that neurologic deficits happen at functionally low levels, at levels that are within the normal range, but not so terrible that they meet the lab low ranges. So we need to change our ranges to be more in line with what functional medicine has been practicing. Revisiting the definition for healthy B12 levels could promote earlier intervention and prevention of cognitive decline. Well, boom, if Alzheimer's disease is the 7th leading killer worldwide and projected to be 1 million people a year in the US alone by 2050, preventing cognitive decline would be huge financially for the country, quality of life wise, all of that stuff. So figuring out what the right level is and nailing that is going to be very important. Inadequate amounts of B12 could induce neurological deficits at a threshold that is higher than the one in current use. And our current understanding of optimal serum B12 may have to be revisited to account for the clinical manifestations. So again, saying it multiple times in a different way there, we must change our levels. We must put out new levels that take this information into account so that we can prevent early cognitive decline, preserve brains, preserve neurons, preserve spinal cords, preserve red blood cells, preserve detoxification, preserve all the hundreds of enzymatic reactions that B12 is involved in for global physiology. Let's open this up for questions or comments.
Can you please repeat the best test for B12?
Yeah, it's called methylmalonic acid, MMA for short. But right here is the spelling, if you want it, right here. And there's a serum level and there's a urine level. This paper used the serum or the blood level.
Just incredible that you just keep getting proved right with the levels.
Yeah, it's just more validation, right? And
Right.
so if it's funny because they say, functional medicine doctors will say, we get these results and all that. And, mainstream will say, well, you don't have any studies or, randomized controlled clinical trials. It's like, yeah, but we have tons of clinical data. And then here in the paper, they're saying we shouldn't be going by these arbitrary ranges. We should be going by clinical observations, right? It's like,
Yeah.
So again, that validates it too. That doubly validates us. We're saying, hey, you're using the wrong ranges. We're using these ranges and getting results. And they're saying you're quacks. And then the papers are saying, oh, well, actually, the quacks have been doing it closer to right than you have. But yeah. Let's see. Kathy says, I had a portion of my GI removed that supposedly absorbs B12. I used to take a monthly shot for years until my insurance nixed it. Have you heard that a portion of your intestine is the only way to get it, not along the entire intestine? Yeah, there's different absorption areas. And so that's why, getting the B12 shot subcutaneously could be helpful for people who aren't absorbing intestinally. And you could try sublingual, and because you can skip the GI tract that way and hopefully absorb it through the oral blood supply there. And then another thing that's widely available now is the different, medical spas that do IV, and you could go do an IV drip of, B12 or B complex, or there's all kinds of cocktails you can do. So if the insurance won't pay for B12 shots, that's, doesn't limit you as much as it used to. You could try, most inexpensively would be a sublingual B12, supplement. And then if that doesn't move the levels and or the clinical case the way you would want, then you could try, a medical spa and do they have the subcutaneous B12 injections? Many of them do. And or do they do IV therapy? And those are all over the place, at least in the Charlotte area.
There's one across the street, actually. So, yeah.
Yeah, there you go.
Thanks.
Mm-hmm. Was there another question?
Well, now you have me concerned because I'm looking at some blood work and MCV is high and MCH is high. And when I asked her possible reasons, she said your B vitamins might be low or possibly H. pylori. We checked for H. pylori and that was negative. And I had a B12 shot.
Well, so the B12 shot might have taken you into a level that is in the normal range, but maybe for you isn't enough. So there's that possibility. Another possibility is the MCV, MCH, those guys are not necessary, like they can represent B12, but they also can represent folate, which is B9, vitamin B6 as well. So it might be one of the others and or B12. And then your homocysteine. So you asked about the methylmalonic acid. The homocysteine also is when you see those MCVs elevated. My question is, what's this person's homocysteine? Because the MCVs elevated could suggest again the B vitamin issue, which could suggest a methylation issue. And if you're not methylating well, then your homocysteine will build up. And again, that B12, that B9, that B6, those are key cofactors for the genes in the methylation cycle to produce the enzymes we need to recycle that homocysteine and turn genes on and off. And there's plenty of videos on my channel on that. All right. Well, thank you for everyone being on and participating. And I hope you all had a wonderful Easter. And we'll talk next week.